Today, we will be looking at a study that was published in November 2018. This study was conducted by the Micron School of Material Science and Engineering at Boise State University in conjunction with the Idaho National Laboratory. This study used transmission electron microscopy, or TEM, to analyze nuclear graphite. In nuclear reactors, there are three different types of moderators used. There's light water, heavy water, and then nuclear graphite. These moderators in nuclear reactors are used to slow neutrons that are ejected from the nuclear fuel rods. These slow neutrons then react with the nucleus to induce nuclear fission. At this time, it is impossible to analyze the irradiation damage that is caused to nuclear graphite in these nuclear reactors. TEM offers an alternative method to looking at the atomic level deformation that is happening. TEM, however, uses electrons as opposed to neutrons, which are causing the damage in the nuclear reactors. To help us better understand how to look at the study, let's take a look at what TEM is and how it is used.
TEM is used to study nuclear graphite, since neutrons damage the material. The electrons fired at the material do not react nearly as much to the sample as neutrons do. TEM stands for transmission electron microscopy, meaning that electrons are transmitted through the sample and onto the screen. The electrons come from an electron source and are transmitted through a condenser beam system onto the specimen through a bunch more lenses until finally appearing onto the screen for scientists to see them. The image on the screen now is what a sample might look like on an atomic scale from the paper we are discussing. The lines you see here are where columns of atoms are formed together to make up the material. As you can see here, TEM has the capabilities to show extremely fine details in the crystal structure, such as defects. Samples used in TEM imaging need to be prepped in a precise way in order to be usable for imaging. Now we will go over general sample prep for TEM imaging.
To ensure imaging can be completed prior to sample destruction, careful sample preparation is required. Sample preparation varies depending on the type of sample being imaged. A lot of steps are required for successful sample preparation. To prepare conductive material samples like metals, electro or jet polishing is required to produce foils. This can be done through the use of an automated electropolisher. During this process, the specimen is made into the anode of an electrolytic cell, so an applied voltage dissolves the metal and deposits it onto the cathode. This thins the center of the sample until a hole appears in the middle. The remaining material around the hole should be thin enough for viewing on the TEM. The resulting disc should be about 3 millimeters in diameter. To prepare a ceramic sample, you take a thin slice from a material bulk with a diamond wafering blade, then polish one side and glue it to a glass slide. Grind it with silicon carbide paper to about 100 micrometer thickness. Ultrasonically machine a 3 millimeter disc with an ultrasonic drill. If none is available, this can also be done by hand. Dimple your piece using a polishing paste and rotating wheel to create a bowl-shaped recess onto one side. Then ion beam milling utilizes an electric field concentrated and accelerated through an aperture, shooting the beam through two guns onto both sides of the sample, thinning until a hole appears, leaving the material around the hole thin enough for TEM viewing. Organic sample preparation can be done through chemical or freezing methods. We are only going to focus on the chemical method. Fixation is done by soaking and or washing the sample in different solutions of chemical preservatives, then it's rinsed in distilled water. Next, the sample is dehydrated in stages by soaking in ethanol or acetone solutions. The sample is then embedded in a polymer resin epoxy, cured until polymerization is complete, and then the sample section is cut thin with a microtome. Then that's collected onto a copper support grid and coated with carbon. Now your samples are ready to be examined. Now that we've covered sample preparation, we will cover the specific TEM techniques that were used in this paper.
This study utilizes TEM techniques that should be familiar to us having taken MSE 321. These include sample preparation, high resolution, and as we learned in class this semester, bright field TEM utilizes objective apertures to block the diffracted beams from going. The micrographs below were produced by the authors of this study using high resolution TEM. High resolution allows them to achieve a very small scale, 5 nanometers, in analysis, which allows a better understanding of what's going on at the atomic level of the material. The left two images received a minimal electron dose, and image A, at room temperature, you can see very little damage in the carbon structures. Image B was done at 800 degrees Celsius, and there is some swelling in the basal planes as well as some curling. On the right side, we have an image that was produced on samples that received a high electron dose. In conclusion, we hope we have demonstrated what TEM is and how these were applied in a science. Thank you.